Like into one lecture that kind of self-assembles itself, so hopefully it won't be too scattered. But I think I will raise more questions that I can answer in one lecture, but we should promote some very nice discussion overall. So this is basically some of the work we're doing at on multi-scale simulations of bio-inspired and biological materials, and in the sense that we're looking at things at different scales. We're looking at some of the quantum level effects in materials, such as reactions, but we're also trying to look at larger length scales and understand length scales and things that happen beyond the atomic scale behavior. So this is some of the work we've done along those lines. And biology is very interesting. There's a lot of different sources of inspiration for us that we can get from biology. These are some of the problems that have been mentioned and also are of interest, like separation of particles. So for instance, selective transport or filtration applications. There are many interesting ways biology takes care of these kind of problems. For instance, ion channels are very effective in allowing certain particles to pass, certain atoms to pass, other molecules to not pass. And this is quite interesting in biology, and it's a very well-developed area of interest. There's other applications. I talked a little bit about protein nanotubes, and you heard about carbon nanotubes as well, and applications in drug delivery or toxicity, these kind of problems, I think.
Sorry, just a question. Before you mentioned something about the bending stiffness of the triangular shape.
Yeah.
It is maximum in some sense.
Yeah.
Can you explain what you?
So basically, when you have a triangular system, it maximizes the amount of area you have away from the center for isotropic shapes and polytopes, like closed forms. It's the best shape in terms of bending rigidity per area.
Bending rigidity per area.
So like a radius of gyration kind of approach.
Is this? I have to think about this.
Yeah. I mean, it's very complicated actually, because I thought a lot about it too, and I thought circle would be the best.
In fact,
what we usually learn, but when you look at the calculation, the triangle actually works better. But when you look at other aspects like circumference, etc.
But for bending, there are probably some other constraint.
Yeah.
For bending, the further away from neutral axis, the better mass. So maybe there may be some constraint here. The cross-section area is given. What about total amount of material is given?
Yeah. So cross-sectional area is what you have as constant.
Okay.
And then I have a picture of it actually. Basically, this is equilateral triangle.
Possible?
So if you have the circle versus the triangle with the same area, in the case of the triangle, you put more area away from
okay.
But
same area contained by the so not same area of the cross section or not?
Cross-sectional area. Yeah.
Cross-sectional area. The material, I'm referring to the material.
Yeah, basically material volume in that sense.
But what about distributed material as far away as possible, keeping the same, just like a slender triangle?
Okay. So
what about those cases?
So in that case, you lose you need to go into concave shapes. So that's harder to treat, I think. So we haven't looked at that. But if you look at convex shapes, triangle is the maximum.
No, just a slender triangle. Slender rectangle.
Perhaps it's just with maximal inertia.
But
only one
in
direction.
all directions.
Oh, in what direction?
Yeah, so that's the buckling is the weak axis that gets.
Okay.
It's either circle or triangle. So and those are two limits.
Okay.
So depending on how you take the constraints, one becomes better.
Okay.
In the area constraint, triangle is the best. But the other issue is the roundness of the corners. So the more round the corners become, the efficiency starts to drop. So for the structures we looked at, the roundness I think is in the range such that the triangle is still better. But in many cases you can't make them very sharp, so then the efficiency goes down. This is just a general description. There are also other issues in continuum mechanics where we have localization of stress in the edges and so forth. So we don't use it as much as circle structural elements, but mathematically it's a limit which is of interest. And for soft matter, it may be actually a better option than certain. But this is a hypothesis essentially. We don't know why it is that way for sure.
Strange.
Very counterintuitive, I think. So.
Yeah.
Okay, so yeah, basically that's the story of the active phase T4. And then also we are looking at other types of materials where you have certain aspects of multifunctionality, so certain structural aspects that can vary when you change the chemistry. And we talked a little bit about hair and how the chemical structure of hair can be changed with chemical signals, which leads to different kind of mechanical features or structural features that are of interest. So these are some of the problems we're currently looking at. And I will go back to the case of spider silk and then expand on all of these different problems, kind of drawing analogies between different structures. So in the case of spider silk, in the last lecture I talked about these nanocrystals, beta sheet nanocrystals, but I really didn't discuss anything about the amorphous phases. So now I'm going to talk a little bit about the amorphous structure of silk and how that plays a role in the overall mechanical behavior. Going back to what we discussed, we have two phases. Actually, it's more complicated, but let's simplify it as two phases. One is these nanocrystals, the other is the amorphous regions. In the amorphous regions, we have basically glycine repeats, which are basically either thought to be 3-1 EDCs or beta spirals, so spiral type shapes, or some kind of a helix shape that's similar to alpha EDCs. But and these are hypothesized to provide extensibility and toughness to the material, but their atomistic structure has not been discovered because it's amorphous. It's very hard to characterize with regular experimental methods. But we do have some guidelines on what kind of structure these could be. So the idea here is to basically map something like a representative structure for the case of amorphous domains. And then basically the idea is to link all the genetic information we know from spiders, such as the sequence of the proteins, etc., and kind of bridge that into a structure, and then from that structure get some sort of mechanical features. Some of the interesting things here is that if you look at all the structural materials, structural proteins, like elastin, resin, collagen, muscle, etc., not care about necessarily, but protein-based materials. The key thing there is a lot of these materials have glycine repeats, and this is almost conserved across many different structural proteins. So what I'm going to talk about next might be very relevant to other protein structures as well. So in the case of spider silk, we have dragline silk. We have two different kinds of proteins that are very dominant, and this is both of those are very rich in glycine repeats. So one is GGX, where X can be 1 or 3 or 4 residues, and the rest is glycine. And the other is GPGGQ, which is glycine protein and then glycine and then other amino acids. But a lot of glycine essentially when you compare it by weight. What we try to do here is basically start with the input sequence and then run some calculations to predict the structure. And then you get an ensemble of different configurations of the amorphous and crystalline domains. And then we do some comparison with experiments, and then we do some mechanical characterization. And the idea here is basically we need to do a very large, either a very long simulation or a very different kind of sampling approach to be able to predict the structure, since this is a process that takes time. And what we've done here is what's called replica exchange molecular dynamics. So you basically run the same replica of the simulation at different temperatures, and then you do basically Monte Carlo type exchanges between the different temperatures. So at a high temperature, you might be sampling through many different configurations, and you might find a low energy system. And then once you find that low energy system, it basically diffuses down the ensembles into the low temperature, so that you can do a more careful characterization at a more realistic temperature. And the idea basically is you convert the folding problem into sort of a diffusion problem across your processes, which is much more efficient than just trying to run normal simulation, where there's a bigger chance of getting stuck in one state. So it's almost like jacking up the temperature to go across this, but you're still at a low temperature with the characterization. So from this we get a very large number of different structures, and then we do statistical analyses on how these structures may be relevant to the actual structure, which we don't know. So, okay, we've done this, and in the end what we get is basically representative self-assembled structures for the crystalline and amorphous stages of the proteins. What we observe here is that amorphous regions have disorderly structures, conformations can coexist in many different configurations. And in the end, we get a listing of what could be the most probable structure. So we look at the similarity between different structures by looking at the coordinates of the atoms. And then we can say, by looking at all these thousands of different configurations, what kind of configurations are most likely from an energetic perspective and also a likelihood perspective. And the key findings here is, as we expected from experiments and also our simulations before, the polyalanine repeats, or the glycine-alanine repeats, typically form these crystalline domains which are very orderly. And then in the vicinity of these crystalline domains, you have these disorderly domains, which basically represent the amorphous or semi-amorphous phase. And this is looking at two different proteins. In the case of the GGX protein, you have more of an orderly structure, it's almost semi-crystalline. But in the case of the protein where you have more prolines, the MASP2 protein, it's less orderly. And I'm going to explain next why this is the case. So we also looked at what we call Ramachandran plots, which is related to the actual structure of the system, basically looking at the torsion angles of the protein and for specific amino acids. So we looked at glycine and alanine, which are the two amino acids that create most of the spider silk, and we looked at the angles of each of these cases. In the case of alanine, you see a very clear peak in this region at the upper left corner of the two angles that represent the structure. And this corresponds to these beta sheet crystals, and this is very well known in structure characterization. In the case of glycine, glycine is a very flexible kind of chain. So it's a very small amino acid. So what you observe is that it's got more of a spread, but basically you still fall into the ranges which have been defined experimentally as well, which are essentially similar to beta sheets, but slightly different in terms of angles. So that's the next thing we need to explain. Experimental findings tell us that in the case of glycine, we have basically a repeat unit along the chain, which is 9 to 10 angstroms for 3,1 DDCs, which have been proposed for some of these structures. And there's some relevance of this with respect to the mechanics, which I will discuss next. And then when we look at the evidence from atomistic simulations, we also look at the probability of every third amino acid and how different the spacings are going to be. And when you plot that probability distribution, we find that every third amino acid is in glycine is separated by approximately 9 angstroms, which is also in line with what is observed with the experimental results. So the basic bottom line here is you have two peaks, one is around 9, the other is a lower peak around 5 to 6. And this confirms that the GGX repeats we have form these long elongated structures which are semi-extended, oriented, and sparsely hydrogen bonded. So it's not like the crystals, but it's somewhere between fully amorphous and fully crystalline, which is related to some of the experimental observations. In the case of the prolines, proline is a very unique amino acid. It basically creates a turn in the sequence of the structure. So it's very much related to proteins that have sort of a coiled shape, like collagen has a lot of protein as well. So the result of that is basically, if you go back to these pictures, so the second column of pictures, you can see that because you have these prolines, it's very hard to crystallize into a regular structure. Even if you apply a stretch, you have to apply a significant stretch to be able to crystallize these domains. So that works as what we call a beta sheet breaker. So it basically limits the domain of these amorphous regions and the crystalline regions. The crystalline regions to nanometer size. And as I mentioned in the previous talk, this is important because if you have very long crystals, cooperativity of the hydrogen bonds don't work as well. So this is the basic purpose of probing, we think, basically creating these amorphous domains and also limiting the size of the crystalline domains, such that you can have a clean two-phase kind of material. The role of the first phase, which is still semi-extended and oriented to a large extent, is related to what we call the pre-stretched molecular configuration of spider cells. So as you draw the spider silk, you basically align the molecules and you have a semi-crystalline phase which gives a high initial stiffness to the material. So this is what we think is an important aspect. And then we can take the most representative structures we have and basically stretch them. And as you stretch them, then you can see clearly that there is more crystallization occurring due to the formation of new hydrogen bonds in between the strands. And this provides higher stiffness as you stretch it more and more, plus you're stretching the covalent bonds more, and therefore you have a high nonlinear kind of behavior. This is observed to a lesser extent in the case of proline, which again causes breakage of the crystalline domains. The key observation here is when you look at these chains, and basically maybe pull half of the chains this way and half of the chains the other way, what you observe is you again have this sliding kind of motion, which relates to how the hydrogen bonds breaking the crystal and basically the material yields through the breaking of the crystals. So initially stretching the amorphous domains and then yielding at the crystalline domains is what we observe. Okay, so the mechanical behavior is basically can be characterized by several regions. In the initial region, you start off typically stiff, and then as you break some of the bonding in the amorphous domains, you have sort of a tapering, and then when you come to the third stage, when you're pulling the covalent bonds, it becomes stiffer again. And you can characterize these by looking at the structure while you're stretching it. So you can look at the number of hydrogen bonds that I have in the system, or the number of turn structures. And as you stretch it, of course, you have less turn structures. And initially, you get a decrease in hydrogen bonds, and then you get a slight increase in hydrogen bonds as it crystallizes as you stretch it. And then finally, once you start shearing it, you get a drop in the number of hydrogen bonds. So we can really characterize different phases of deformation in these materials. So, okay, most of the structures I showed in the context of spider silk takes advantage of these two domains, which is one is crystalline, the other is amorphous, as I mentioned. And I basically started working on a more recent project where I can draw some analogies with respect to the spider silk structure, but now looking at more of a multifunctionality aspect. So in this case, what we're doing is, instead of having these solid crystals, could we have another kind of structure that is basically, again, crystalline and has a hydrogen bonding, but instead has a hole at the middle of it, such that you can have a nanoporous membrane. So you could have basically a tough membrane, but that could also function as a filter. And this is important for many different applications. You could use these for gas separation, or ion channels, or in fuel cells, and so forth. But one of the key criteria is that it has to be a fairly strong material that is resistant to degradation. In pursuit of this goal, we're collaborating with Tim Xu and Brett Helms at the University of California, Berkeley, and Lawrence Berkeley National Laboratory, who have actually devised a way of really creating these membranes, which is, I think, quite fascinating. The process is as follows. So what you have here is basically a protein structure that doesn't really exist in nature. So in nature, most of the proteins we have is a left-handed protein. And then by playing around with the chirality of making these mirror images of the amino acids, what you can create is this peptide that kind of creates a circular shape. So these are called cyclic peptides. They basically just create ring-like structures. And when you do that, the nice thing is, as you can see from this picture, you can have hydrogen bonding in between these rings, and that can cause the self-assembly of these nanotubes. So you can form micrometer long nanotubes that get self-assembled, so different from carbon. And this was demonstrated by Galili in a very famous Nature publication in '93, and then has been replicated for different kind of protein structures. The key thing to realize here is that the core of the nanotube is really in the order of a nanometer or even less, so it's really, really small. That individual nanotubes have been observed for a long time, but they haven't been really used in membranes much until very recently. The reason is once you form these nanotubes, they are fairly stable, so it's very hard to process them and make a membrane out of them. So what the Shu group and Brett Helms did was come up with a method where you can co-assemble the nanotubes with some basic polymers, block polymers, and basically have a system where you have the nanotubes dispersed within the material, extended throughout the membrane, and basically vertically aligned and with more dispersed kind of nature. So this is the main experimental breakthrough that leads to our interest in looking at these materials from a mechanics perspective. So the key idea here is first understand how the mechanics of these nanotubes behave, like how stiff they are, what's the persistence and these kind of things. And then the second is look at the transport behavior through the nanotubes and understand how we can use these for different kinds of applications. And this is essentially the first example of organic nanotubes fabricated into a functional membrane over macroscopic distance. So you can really make thin films out of these and hopefully use them for technological applications. Okay, so going back to what we had with the spirals of nanocrystals, we tried basically the same kind of thing where we try to pull one strand out and look at the deformation mechanisms and favor approaches using steel dynamics. And first we of course looked at the thermodynamical stability of the system, and what we observe is that the number of hydrogen bonds and the overall stability of the molecule is fairly consistent. So these are these do assemble into these and then we next looked at deformation mechanisms when you apply a force to the center of the nanotube. So in this case, mechanical resistance again comes from interstrand hydrogen bonds, which can break due to stress, random thermal vibrations, and also attacking of water. So water also wants to make a hydrogen bonding with the backbone. So it actually decreases the stability of these particular nanotubes. And this is also known for the case of spider silk. In the case of spider silk, though, you have these hydrophobic side chains which push the water out. So you have almost a phase separation from water, and that plays a protective role for the case of the crystals. And then when we look at the deformation, we can do some very simple analyses. And what we observe is basically failure typically occurs in what looks like a hinge formation. So instead of just shearing the system, you need to break it at 3 different points, which basically forms 2 hinges and then the central point, which leads to stability. So an alternative mechanism, if you had a very stiff nanotube, would be localized failure in one or the other side due to some heterogeneity, or localized failure at the central strand which you are pulling on. And that can be observed. So we've looked at again different rates, looking at 4 orders of magnitude. But the rates you need to be able to do that, to basically pull one strand out, is much faster, I think around 25 meters per second, which for the cases of membrane applications is way too fast for such a thing to occur, unless you have a shock loading kind of scenario. So what we think is going to happen is more of this kind of a scenario when they break in water. Of course, this might vary when you co-assemble them in a membrane, so it's sort of something we need to look at in the near future. Okay, so the idea here is basically we didn't observe the stick-slip type dissipative behavior. This is both due to the deformation shape of the molecule, but also it has to do with the cross-section of the molecule. So in this case, we have more of a tubular section as opposed to a crystalline stacking of strands. And that's an important consideration, because if you think about it thermodynamically, rebinding does play a role on how easily you can break the system. So if rebinding is not favorable, then you're jumping to a state which is easier to break. And in the case of a tube, you can look at cross-sections of two different structures. The rectangular case of a crystal versus a tubular scenario. And in the case of tubular scenario, if you deform it just a little bit, it's much more likely to lose its rebinding capacity, because the overlap area diminishes very fast here, whereas it's much more gradual in the case of rectangular areas. So that's basically all we have so far on mechanics of these nanotubes, and we're doing more analysis of trying to characterize the rigidity and persistence length factors, which we can compare with experiments. So that's one thing we hope to do in the near future. Yeah, so the bottom line is the considerations for transport and mechanical strength of the membrane are sort of disparate objectives. I mean, you want tubes to be able to have porosity, but on the other hand, it seems that if you have strands, you get more overlap and more capability for energy dissipation. So this is something we need to think about a little bit more and kind of figure out what would give us a tubular structure, but still a very high mechanical strength. And that's, to a large extent, chemistry dependent as well. Okay, so, and then I'm going to talk a little bit about assembly and transport considerations for the case of these nanotubes. So I mentioned that these nanotubes are really, really small. So it's basically the size of a nanometer or so in cross-section. And when you look at this, you might wonder, okay, what kind of a flow behavior can you have with such a small nanotube? And there's a lot of literature in this area, because there has been a lot of interest in looking at carbon nanotubes for these transport applications. And the idea is basically, as you make it smaller and smaller, water begins to get a more orderly structure due to surface area, so it's almost like a crystal. And at nanometer dimensions, for instance, carbon nanotubes, water typically flows in a single file flow. So it's one by one, kind of like a train-like structure with a specific hydrogen bonding pattern. So it's interesting because if you think about a carbon nanotube, you have basically a hydrophobic surface. So the water doesn't like the carbon nanotube surface. As a result of that, you have what we call sort of a slip-like flow behavior. So slip length is sort of a continuing description of how much friction you have in the channel. And in this case, you have much less friction than what would be a stronger chemical interaction with the water. So that's interesting because you can have very fast flow through these nanotubes if the water doesn't want to be in there. The question there is, okay, if the water doesn't want to be in there, well, how does it get into the nanotube in the first place? Right? If the system is hydrophobic, I would expect the water to stay out of the nanotube for the most part. Well, there's many theories as to how this occurs. One of the theories, which is I think probably the most widely accepted so far, says that the average free energy of bulk water should indeed be lower than the water confined in a hydrophobic channel due to the possibility of making more hydrogen bonds in the bulk. But you observe that there's a lot of water molecules that really don't have this kind of hydrogen bonding, which has a higher energy. So that some of the molecules are not fully coordinated with other water molecules. And as a result of that, you get random events where you have a water molecule getting into the nanotube and forming a coordinated system there with a hydrogen bonding with adjacent molecules. So since the energies of the two cases are fairly comparable, overall, on average, you have water molecules filling in the nanotube. And this is a model that was developed by Hubbard and others earlier in 2001, I think. So what we observe from this behavior is that we can have very fast transport through the nanotube once it fills in, and in many cases it has been reported to be orders of magnitude faster than continuum level bulk diffusion experiments. So the question here is, can this be quantified with molecular dynamics, and what kind of parameters govern this flow behavior? So, and this has been done in the past quite a bit heavily studied. People have looked at scenarios where we have basically no pressure applied to the nanotube. So what you observe here is there's no pressure applied, and you might have a chain of molecules that basically get in, and then they will hop back and forth, and then eventually they go out from one side. And that counts as one flow event in one direction. And then you might have the same kind of flow occur in the other direction. And overall, the number of flows in one direction versus the other are equal, because there's no pressure directed in one direction. But this basically is a nice scenario where you can get an understanding of how fast we can have flow through the nanotube without the issues of applying the pressure externally to the nanotube. So these are considered counting flow events in one nanotube, even though there's no net flow. Okay, so the basic idea we demonstrate here is that the number of flow events per nanosecond can be counted and compared for different parameters. And there's many studies in literature with regards to these flow measurements. And if you look at all the literature, there's many different factors that play a role. Nanotube length obviously plays a role. Single versus arrays in membranes have been used. Slight variations in force fields are observed. Different thermostats, so different ensembles, different boundary conditions, and different time scales of simulation are some of the factors. So if you want to really understand how this flow events occur, we really need to understand what are the real factors that are variable, but that really matter. So that changes the flow rates or flow count events. So we did some studies along these lines just to get a better idea. And from our observations, there are two factors that really play a crucial role. One is the nanotube length, which is very intuitive. I think this is something we expected. The other is different thermostats, which we really didn't expect as much. I mean, it's not the first thing that comes to mind, but it does seem to play a major role. So the nanotube length effect is very simple. As we have a longer nanotube, it becomes less likely for this random flow event to occur, since you need to push more water atoms in one direction versus the other. In the case of the ensemble, it's a bit more interesting. So what occurs here is that you need an ensemble to be able to modulate the temperature in your simulation, so that you have a constant temperature system. And this is done in many different ways. One of the ways you do it is basically by adding a damping term into the simulation such that extra heat can be taken out. And when you do this, what you observe is that the damping kind of changes the flow events quite a bit. So you can have almost an order of magnitude when you use the typical values used in literature. So it makes it very hard to compare, let's say, a scenario with high damping versus a scenario with low damping. And this seems to play a major role in our interpretation of the results. And then you can also have a dependence on the temperature as well, which is expected from any kind of diffusion scenario. So the idea here is basically comparing all these temperature effects and damping effects. At the bottom line is if you thermostat the water, you break the local velocity correlations within the nanotube, because you're manipulating how the molecules are vibrating and moving with respect to time. And then reducing high velocity motions in the overall system are sort of important in triggering and sustaining these unidirectional bursts of flow. So this we think is probably how the mechanism plays a role, and how a thermostat can change that mechanism to some extent. So just an interesting concept. And then even the issue of length dependence is not actually that clear for these nanoscale flows. This is a fairly complicated problem. There's also different findings in the role of the nanotube length on different scales. So one of this is one study basically shows us that there's a length dependence, which is fairly strong. This is the length essentially, and this is the flow rate. In another study, we have basically no dependence on length. And this also plays a role if the flow is basically pressurized. If you have pressure-driven flow versus no pressure. In the case of pressure-driven flow, you have less of a length dependence, probably because you can go over these barriers that the length induces very easily when you apply energy into the system. In the case of this scenario, you basically have a lot more length dependence. So this is something that we need to look at more. And then as we go into membrane applications, length really becomes a key, because you can't have a too thin membrane. It doesn't have mechanical strength to function as a transport medium. And then if you have it too long, then you might be susceptible to reduction in the flow. So no one has really looked at anything beyond a few nanometers, so this is something we hope to look at in the near future. And then you also have dynamic vibrations and other factors that will play a role, as well as global stress conditions, such as pressure that causes a tension in the membrane direction as well. Okay, so from a gas separation perspective, the ideal membrane would have to have certain desired qualities. It would have to be stable under a wide range of process and service conditions, so it should be chemically, mechanically, temperature-wise very stable. It should have a very accurate selectivity for small differences in chemical properties, so such as polarity and size. And then it should have a continuous vertical orientation, and then has a large flow rate with a very small driving force, and then also at a low cost if possible. But this is a lot of constraints, of course. And many people have looked at this application of carbon separation, which is really important for many different industries. One example is natural gas industry. If you have in the natural gas pipelines, carbon dioxide, this causes corrosion. So there's a lot of interest in trying to separate methane from carbon dioxide to reduce corrosion, and also increase the energy efficiency when you burn fuel. So there's many different options that have been proposed. Inorganic nanoporous materials, hypercrossing polymers, which are low cost, but they're very large pores, so they're not very selective. And then carbon nanotube membranes have also been proposed, but it's very hard to get vertical orientation with these. And the length and pore size distribution and chemical modification of the core are some of the challenges in that technological application. So the idea here is basically co-assembling these cyclic peptide polymer conjugate based organic nanotubes with some polymers to create these subnanoporous films. So I mean, you can look at many different kinds of scenarios of flow, and one of the simple things we tried to do was basically see if you have a few dispersed molecules of carbon dioxide, whether they're going to flow through the nanotube. They do enter the nanotube spontaneously, even though they are very loose. But we didn't see a very clear flow event in this small nanotube case. So we're really looking into how we can understand if you have a high pressure system or different boundary conditions, how this flow is going to change, or how this transport phenomenon can change. One thing that is of interest is, of course, this is very different than the case of the carbon nanotubes. You can see that this system is basically fluctuating quite a bit more than the case of the nanotube because it's weaker one. So this is going to play a role in the transport phenomena as well. This we call the breathing modes of the nanotube. So it's basically solid mechanics, fluid mechanics coupled with multiphysics kind of a problem. Okay, so the key challenges here is actually quite counterintuitive to me as a mechanician, because what I thought was initially that it is good that we study sequences that are structurally stable, that assemble rapidly, but in terms of polymer processing, if you have very stable nanotubes, this is a bit of a problem. So the reason for that is the nanotubes formed prematurely when you're trying to co-assemble them with the matrix. So you don't have a very dispersed, co-assembled, nice, uniform structure. So in this sense, strength is not, unfortunately, not always a plus. So we can't just design for strength in this case. And we need to basically come up with some chemical modifications to the nanotube such that you maximize selectivity, you maximize transport rate, and also have very good co-assembly methods while minimizing structural fragility. So this is the whole kind of the optimization that needs to be done for the specific case of thin membranes. And the approach we're taking here is a computational screening of different chemical modifications to quantify the effects on the mechanical rigidity of the nanotubes and then also the transport effects. So there's several studies that have looked at how we can modify these nanotubes. So these are so-called mutations that you can do to the nanotube. And one of these is what has been proposed by the Helms group, which is basically you put in a ring-type structure here that has a methane group, that methyl group that points inwards. So when you do that, you can change the size of the pore quite significantly, and the shape of the nanotube also changes. So the implications from a mechanics perspective is that first you're varying the inter-ring binding energy. So instead of having this kind of a radial structure, which can form hydrogen bonds very easily, now you have this bulky system here, which can still form hydrogen bonds, but basically adds a heterogeneity into the system. And then the second thing is you induce a ring strain, which causes a non-circular shape. So the bending rigidity of the nanotube becomes irregular and different as well. And then the third issue is, of course, we change the internal chemistry and the pore size. So the transport phenomenon also will be quite different in the case of these kind of nanotubes. The other thing to note is that this is extremely difficult to do with a carbon nanotube. You can't really easily change the internal chemical structure. So that's one of the key advantages of organic nanotubes. So using market dynamic simulations, we really try to understand what the real effect of this chemical modification is. And in this sense, when you look at these regular nanotubes that I showed earlier when we were doing mechanical stretching tests, these are fairly stable. I mean, you have a consistent number of high number of hydrogen bonds that basically allow stacking of these nanotubes. In the case of the modified ones, depending on the chemical structure, you can have very different behaviors. So if you have protonation of the side chains, you have more electrostatic repulsions, which combined with the weaker hydrogen bonding interaction, reduces the binding energy of the nanotube. In the other case, you can have much higher hydrogen bonding, but still much lower than the case of the regular nanotube. So it seems that we, by playing around with the chemistry, we can really get the optimal kind of scenario where it's a bit weaker than the regular nanotubes, but it is still capable of self-assembly characteristics. And this is the same kind of thing that has been observed experimentally for these systems. And then we can dig deeper into the simulations. Since we have these trajectories, we can really look at what's going on. And what we observe is that we're interested in which parts of the hydrogen bonding changes when you look at this structure. And what we observe in this case is that in the vicinity of the modification, we have a variation of the hydrogen bonding pattern. So it seems that we have a consistent way of changing the chemical bonding. And then the other thing is, of course, the pore size. How does the pore size vary as we modify the nanotube? And here we can see that this is basically 4.7 angstroms in width, whereas in this case we have more of a wider nanotube. This is a regular nanotube. So now we're coming into the scales where you can really differentiate between very small molecules like carbon dioxide versus methane or other kind of molecules. And then by changing the chemical structure here, instead of the methane group, we could have let's say a polar group which could make the inside polar, and then you can have non-polar particles slip through the nanotube. So you can change the slip length for the continuity of slip. Okay, so the idea here is just to show some of the very preliminary things we're trying to do in terms of bridging the landscape. So if you think about the whole problem, what we have to do here is model the membrane, and then model the nanotubes, and then also model the transport issues within the nanotube. And these are sort of coupled events. You have basically a tension in the membrane that can change the porosity of the membrane, and this can be used as a function to tune when you want the molecules to pass through. So by opening or enlarging the ring by applying mechanical tension, you can switch it on or off in terms of transport capability. So we want to be able to really model this at larger length scales, which is, if you look at it from an atomistic perspective, it's not possible to do. So this is one of the things that we're working on currently. As I mentioned, there's some limitations of all atom simulations. Time scales and length scales are quite limited in this range, basically a few hundred nanometers at most. And we need long time scales for a lot of these phenomena. So can we really keep the same mathematical framework and study larger systems? This is sort of the basis of what we want to do with coarse-grained modeling. And basically what we are talking about here is, we were looking at classical molecular dynamics. Now we're looking at mesoscale models, so basically coarser descriptions of the actual systems that we're looking at. And the basic principles of coarse graining is generally simple in terms of what we're trying to do. It's very similar to reduced order models that we have in continuous scale. Instead of representing every single atom, what we're doing is basically take one amino acid, which is tons of atoms and many degrees of freedom, and reduce that into a cluster of molecules. So this particle becomes one big particle, and then the other particle becomes another particle, and then you can have simple spring and angle type of relations between the particles, and this really makes your computation much easier. The benefit, of course, is you greatly reduce the number of degrees of freedom. You can do much faster calculations. And this has been applied to many, many different systems, and it's a field that is growing exponentially in the past 4 or 5 years in particular. One of the applications I think is interesting is fusion of large-scale lipid vesicles. These vesicles are found in the biology, and this fusion effects are very important for biological processes. So, but each of these vesicles can be up to a micron in size. So this is impossible to do all that. This is why we try to do coarse graining. And there's many different groups that have worked on how to do these parameter derivations. So how do you optimize and validate basic parameters for coarse-grained model? This is really a fundamental question. There are several ways. You can run an all-atom simulation, look at probability distributions of atomic configurations, so molecular versus intermolecular. You can carry out an all-atom simulation or a macroscale experiment, and then you can compute certain thermodynamic properties, and then you can try to match the properties. So this is the other way of doing it. And the third way is you carry out molecular dynamic simulations to assess mechanical properties, and then coarse-grained to match mechanical properties along with thermodynamic or structural features. And then what could be some of the properties to train the system for? It could be structural features, such as bonds, angle distributions, geometries, vibrational frequencies, or mode shapes, low energy states, and so forth. You could also try to fit in thermodynamical constants, so looking at free energy unfolding, solvation, diffusion constants, these kind of things. And then you can also have mechanical features such as bulk or elastic moduli or adhesion energy of interfaces. So this is sort of the basis of most coarse-grained approaches that we see in literature generally. And then if you want to match exact probability distributions, this is very hard to do because what you need to do is basically calculate free energies instead of the actual energies, and and basically have the same kind of distributions as the all-atom system, which can be very challenging. So because you have to account for missing entropy effects, and you have to match the smoother energy landscape to the lower system energy landscape. There are many challenges in this aspect. So I'm going to give the simplest example, since I mean it's a fairly complicated topic, but I think we can give some simple examples to discuss the basic aspects of force graining. So one of the things that many people are working on is force graining of water. You would think that it's probably the simplest thing to start off with, rather than some complicated molecule, but it turns out that force graining water is very hard to do in itself. So this is one of the more recent models that has been proposed, which is the Martini model. It's